Welcome to another session of ASM Live, coming to you from New Orleans at the ASM, that is American Society for Microbiology General Meeting. I'm Jeffrey Fox, the features editor for Microbe, the monthly magazine of ASM, and your host for these sessions. I'm going to be speaking with David Valentine of the University of California, Santa Barbara, about some of the work he and his collaborators are doing tracking what happens with oil spills in the ocean and in the water column. But rather than letting me introduce the subject, I'll let David give you a synopsis. I wanted to remind people who are watching on the webcast that if they want to submit questions, use hashtag ASM 2011, and for people in the room, you can signal you'd like to ask a question. Somebody will provide you a microphone, and then please give your name and affiliation, and we'll have fun with the set of questions. Okay, so David, if you would now give us a brief summary of the highlights of your work, and we can get into the details as we talk back and forth.
Yeah, the area that we're interested in is the deep water. That is the water depths of 3,000 feet, 4,000 feet, where the ruptured well was leaking out oil and gas that got suspended and dissolved in these deep layers. And that elicited a microbial response, and we're trying to understand the combination of the flow of the water and how that impacted the biodegradation of the oil components in the context of the spill.
Okay, and so what are some of the findings that come out of this work? We should talk too about how some of the difficulties and challenges of getting samples from 3,000 feet and knowing that they weren't from 2,500 feet, let's say. But first things first.
So the really key aspect of this has been developing a physical model and understanding where the flow of water occurred and how that related to the injection of the hydrocarbons and the growth of the bacteria. And the primary phenomenon that we are seeing is the movement of water away from the wellhead, the growth of a bacterial population, and then the movement of that water back over the wellhead, where the bacteria are already primed and ready to go to consume the substrates because they're there at high abundance. And this, what we're calling an auto-inoculation, seems to have dramatically accelerated the rate at which many of the hydrocarbons were degraded.
And that acceleration has something to do with the unusual geography of this site? I mean, why would that water go out and come back? I don't understand that.
It is because of the geography of the site. If you look at the northeast Gulf of Mexico, where the spill occurred, it's bounded on three sides. It's almost like a bay. If you're
an underwater bay.
Underwater bay,
okay.
At this depth of 3,000, 4,000 feet, it's bounded on three sides, and so there's no current that flushes through. It's not a rapid ocean current that you might think of. It's not anything like a river. The water tends to slosh back and forth, and it moves and it spins, and as a result, it's very easy for water to come over the wellhead, receive hydrocarbons from the wellhead, move away, come back later.
I see. And so is that important in terms of having the right concentration of microorganisms and hydrocarbons? That it seems as if this slower movement of water would have an effect on concentration, and you would have some kind of gradient of shaping up and then reshaping itself with time.
That is exactly right. And in the times when there were pristine, unimpacted waters that were coming over the wellhead, there was a buildup of hydrocarbons. At least that's what we're predicting, and it matches observations reasonably well, that those hydrocarbons would build up in those waters, and it would take quite some time for those hydrocarbons to be degraded. During that time, they could expose many things in the water. They could expose the continental slope where the land comes up. And then there's other times when the waters had already been exposed, the bacteria had already grown, and that water mass comes and sits over the wellhead. And during those times, there's not nearly the buildup, or we don't predict nearly the buildup of hydrocarbons because they're being consumed more rapidly.
Okay. And tell us a bit about how you go about knowing what you're looking at and getting samples to look at. It seems as if you need some special equipment and some high-pressure gear and all that sort of thing.
There's a couple of components to this, and one is a physical component, and one is the actual sampling component. The physical component of this is actually coming from a whole series of observations and a model that the Navy runs, a Naval Research Lab. It's a full oceanographic model that accounts for where all the water goes in the Gulf. And it's pinned to numerous observations of currents from various instruments and also from gliders and things that are dropped in the water and tracked. And then with that
when you say a model that the Navy runs, you're talking about a computer simulation, or it's a mechanical?
It's a computer simulation.
It is, okay.
And
it's not they didn't build a
it's
a miniature bay or miniature Gulf of Mexico.
No, hard to do.
Okay.
No, it's a computer model that receives daily input from observations. And so it casts backward where the water was at different times. And then with that, one of my collaborators, Igor Mezik at UC Santa Barbara, has developed diagnostics to determine where the major flow paths of the water were relating to convergence of water and divergence. And something he published in Science about 7 or 8 months ago, this new mixing diagnostic that seems to be very accurately predicting where different parcels of hydrocarbons and bacteria were. That's the physical side of it. The other side of what we've been doing is actually going out. I've been on 5 research expeditions, and we've been doing quite a bit of sampling. There we utilize specialized research vessels, if we can find them. And we utilize a variety of instrumentation that we drop over the side of the boat on a conducting wire. And so we can visualize in real time various properties of the water column. We can visualize the temperature and salinity and the density structure of the water. The ocean is stratified. We can also visualize if there are hydrocarbons, certain kinds of hydrocarbons that provide signals on some of the instrumentation. With that instrumentation package are a series of water bottles that go down in an open position. And as you bring them back up, you can send an electrical signal down the wire, and that will close the bottle and take a sample at a given location. And this is a standard oceanographic practice. With 23 or 24 bottles on one of these systems, you can get a reasonable sampling set of waters throughout the entire depth range.
All right, and so you've got, and those are the samples on which you do your microbiology.
Yes, that's exactly right.
And so tell us a little bit, or is it not fair to ask, what you're learning about the microorganisms that did such a great job, at least in the deep water part of the Gulf to degrade the oil.
Yeah. Yeah, so we've published a couple of papers on some of the changes in the microbial community and how rapidly the microorganisms degraded certain compounds. And what we found through both observation and is also predicted by the model we've presented today is that there were a series of blooms that occurred. And they had to do with which hydrocarbons were available, which organisms were most abundant, and who could grow most rapidly. And when a water parcel was initially exposed, there were multiple sets of blooms, boom, 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 where they went after the sequentially harder to degrade compounds. The spill itself was 20-something% methane. It was the single most abundant compound. Hydrocarbons, ethane and propane, were the number 2 and 3. And then there's a series of aromatic compounds and other hydrocarbons. The biodegradation didn't go by abundance, though. It went by which were easier to eat. And so some of the less abundant compounds were eaten first, followed by the natural gases, propane and then ethane, and then later on, methane became the substrate of choice for the bacteria. And so we see evidence of those blooms that occurred over time, over a course of initially a few weeks and then months and then maybe 4 to 5 months. You can still see the residual dead microorganisms that had bloomed on some of these abundant substrates.
And there was no, the dead ones were not inhibiting the successor microbes, for example. It was all working. It worked amazingly smoothly, it sounds like.
Yeah, we don't really know if there was some inhibition from the dead organisms. We don't have any evidence that there was, but we don't have any evidence that there wasn't. So that's a very difficult thing to say. When we try and model that, we assume that the dead organisms themselves become respired eventually and are food source for some other organism.
And then the net product, the biomass that is formed, do you follow that at all, or because it has consequences too for the environment?
Yeah. And so we do as much as we can. In the model, we track biomass very closely. We track cellular abundance in particular, and we follow the carbon. And so we know from the emission of what happened at the well and the timing of that emission, which is now fairly well established through a series of federal government reports. We know how much carbon we're dealing with. And so we know roughly what the partitioning of that carbon was between biomass, between respiration, and a lot of stuff that's spilled out of the cells. And we can then predict really how much biomass we're talking about. We put that in the context of the mixing that happened of the waters, and we get the distribution of that biomass throughout the Gulf. Now, the impacts, that is a totally different issue. We're not predicting impacts. We don't really know what the true environmental impacts of that may have been.
But have you gotten a signal or a pattern from this analysis that, I mean, what does that pattern tell you? Anything unusual about what happened in the Gulf? After all, it's a body of water that has a relatively higher temperature, say, than has been experienced with historic oil spills, say, in Alaska, where the temperature at the surface and everything below it was much colder by comparison. So,
you know, the ocean is stratified by temperature. And so what happened in the deep water happened in the cold, and it happened in the dark. And that set up a situation that was completely unique to oil spills. There was no evaporation that happened in the deep water. It all happened at roughly 5 degrees Celsius, about 42 degrees Fahrenheit. And there was no light, so there was no breakdown of oil by sunlight. And so those processes were all very unique for us to be able to study. And it was gas. You usually don't see that. At the surface, the story was very different. The oil components that didn't dissolve into the water on the way up went to the surface. And there, many of the things that would evaporate to the atmosphere had already dissolved to the ocean underneath. Other compounds then began to very rapidly evaporate. There were still many that could. Some began there was not too much that dissolved at the surface. Others were broken down by sunlight. And at that point, at the surface, it's really a foot race between the different processes of natural weathering, and that is the biodegradation, the evaporation, the dissolution, and the breakdown by sunlight, the photolysis. And so probably those were initially dominated by the physical processes. Evaporation can happen very quickly. There were some atmospheric measurements that showed some of the compounds immediately volatilizing. And then the other processes, the photolysis from sunlight, that's happening more slowly, and then you begin to compete with the biodegradation at the surface.
All right, so let me get this straight. So deep down, we're talking about 3,000 feet or more below the surface of the Gulf. The oil that's coming out of the ground is letting off what we think of ordinarily as gaseous material, as a major part of the mix. And yet, that gas is dissolving in the water and it's staying there and being degraded. Is that right? So is that, I mean, I'm kind of curious about the mass balance here of how much stayed there and how much shot up to the surface.
Yeah, so am I. It's a very good question, and the mass balance is still something that's being determined. There are some things we know, that 100% of the methane dissolved. We know that from several different studies that have been published.
And that's surprising, or
No.
makes sense?
No, it makes sense, and I published an opinion piece in Nature during the spill that that suggested that would be the case and that we could use that to quantify the flux. So it wasn't a surprise that was the case. Most of the light hydrocarbon gases also dissolved along with it. And on a total mass, we're talking about roughly then 30% of the total reservoir fluid that emitted just dissolved in those three compounds. And then the next things that dissolved were the monoaromatic hydrocarbons, benzene, toluene, these sorts of compounds. And there were a few percent of the total for mass balance. You add up all the other compounds that dissolve, there are 20-something of them, and you get another couple percent. So you have 30 to 40% dissolving, and then there's this undefined fraction that erupted from the wellhead and broke into tiny, tiny droplets, either with or without the help of the dispersant added at the seafloor. And those droplets, to some extent, became entrained in the deep water. They're so small that they lack the buoyancy to force their way through the water and come up to the surface. And right now, they're the big question mark. We don't know the fraction of that liquid oil component that ended up in these micro droplets. We assume that it's not a really high fraction, but we don't know that for sure.
And you don't have any sense that they recoalesced anywhere? And I mean, there's been some talk from people studying the the more complex organisms that are decorating the seafloor, that they're carpeted or with oil or some residual material from the spill. Is that not the case, or?
It's not exactly the case. There was certainly deposition of hydrocarbons that occurred. The deposition that's been reported, the layers that were 3 inches thick, many of those appear to be pre-existing layers that had contact, that oil contacted and slightly contaminated. That's an interpretation. None of that is been subjected to peer review yet. So that's what we think. Those layers can be found from before the event. They seem to have come from something else. But in close to the wellhead, there was certainly a lot of deposition. And within a couple of miles, there's a tremendous amount of material that fell to the seafloor. So in some ways, you have to think of this in a spatial sense, where there's a ground zero area of massive impact, where the fluids from the top kill operation, where they pumped all of the synthetic mud, that deposited on the seafloor somewhere around that wellhead, and other hydrocarbons got entrained along with that. So there's that zone that is going to be very heavily impacted. And then we're probably talking about pockets of areas that had varying levels of hydrocarbon exposure, including the beaches and areas in the beaches where sand got contacted with oil and then washed back out to shore, or even got buried on the beaches. So there's these pockets that have been shown to exist in several places, and there are probably some in the subsurface, and the model that we've developed predicts where some of those areas might be.
Okay, and your model, I don't know why I'm sitting here and I have this impression that most of the mixing is taking place in a horizontal plane, but this is a three-dimensional space. How much mixing is taking place vertically? I mean, and is it just that the box canyon effect has quelled that kind of mixing, or? Yeah,
there's not a lot of vertical mixing that goes on. You can get certain amounts of upwelling, and during the response efforts, we saw some areas where there was upwelling and maybe 1,000 feet of vertical upwelling, but none of that ever got anywhere near the surface that we saw. We didn't go into the canyons and look to see if there was a localized canyon form of upwelling, but by and large, the ocean is stratified by density. And the waters at the surface of the Gulf, particularly in the summer, are very buoyant waters. They sit on top. And things get denser and denser as you go down. And so these layers don't have enough of anything in them to make them any less dense than they already are. So it's very difficult to get things to move up and down in the ocean. The timescales are much longer than the timescales of moving laterally.
Okay, and how does that affect the microorganisms? I guess they go for where the sources of carbon might be, and but how are they moving vis-a-vis the vertical space?
Yeah. So one thing that we see, the organisms that responded, The ones that were deep were cold-loving organisms, which you would expect in the deep, and the ones that were up at the surface were not. They were very different organisms, and so you had two different responses of the microorganisms at deep and at surface. The vertical motion is not yet established. There were some flux traps that were set out to look for that vertical movement, but I haven't seen anything that came out on that. That was not my work. So we may know something about the raining out of microorganisms, but we don't know it yet.
That does seem interesting, and so whatever is down along the bottom of the ocean is really well adapted to those temperatures and pressure conditions, and things are going pretty quickly once they're established with a particular source of hydrocarbon, because it's a pretty rich environment even without a massive oil spill, right? There are little oil spills to keep them happy in between human disasters.
Yeah, so there's some seed amount of organisms that are there, and it turns out that the nutrients are probably high enough and all the other conditions are sufficient that the hydrocarbons were the only thing that would have limited their growth, and so the organisms bloomed in response. And don't seem to have been stopped by much. There may have been a couple of hiccups along the way for iron or something else, but nothing else seems to have stopped them except their own inherent growth rates.
And do you know anything about whether those microorganisms were doing anything with the detergent or the solubilizer that was put out on the Gulf, or did that all stay up at the surface?
There were two primary applications of the dispersant, the Corexit product that was used. There were over 1 million gallons that were added aerially at the sea surface, and then there was another 770 some odd thousand gallons that were injected right at the wellhead where the material was emanating out. That material seems to have become dissolved along with these deep plumes. And two of the major components have been shown to have tracked these plumes in both the near field and then in the far field. As late as September, October, we were able to measure dispersant to the southwest of the wellhead and actually near the wellhead as well. And when we scaled that to the oxygen loss, what we came up with was a number that looked like the total amount of dispersant that was added. So from that, we suggested, and this is published in Environmental Science and Technology by Elizabeth Kujawinski as the lead author, that the dispersant itself was not being rapidly biodegraded, that it was, at least the main component of the dispersant, known as DOS, that it was acting as a
It's not named after the software program, I hope.
No, it's dioctylsulfosuccinate, but I thought I would spare you.
Okay.
That was acting as a conservative tracer of the plume as the plume moved, and it itself was not being rapidly biodegraded. Whether or not it contributed is not known. The concentrations were pretty low once you got 10, 20, 30 miles away from the wellhead, so it's hard to see how it could be having a major impact, but it itself was sluggish.
All right. Do we have any questions from the we do have a question from the audience. Can you wait for the microphone, which is coming your way? And then introduce your name and institution.
John Rosenblatt, Mayo Clinic. Was it just fortuitous that these hydrocarbon-degrading organisms were located in that area and were able to be stimulated, or is this kind of a general characteristic of most of these kind of bacteria, or at least those that are found in aquatic or marine environments?
Yeah, I think it largely had to do with the environment. And the Gulf of Mexico, it has a lot of oil seeps, over 1,000 natural oil seeps to keep populations going. There's 4,000 oil wells in the Gulf of Mexico, almost 3,800 something. So it's an environment that has a lot of hydrocarbon. So I think the fact that it has a lot of hydrocarbon, there's hydrocarbon seeping out, that is going to tend to keep a hydrocarbon oxidizing population at a reasonable level. Having said that, I would fully expect to see these organisms along other continental margins, but maybe not as at high of abundance. And if this happened in Brazil, I think it would be a very different story as to what happened. The currents would act very differently. There wouldn't be this quiescent water motion of sloshing back and forth. Things would be taken and shot out down the rapid currents.
We have another question.
Mark Middleman with Exponent. Your very nice work and the work of some of your collaborators in this morning's session talked about a lot of the physical-chemical parameters that were replicated in laboratory experiments, but one thing that wasn't mentioned or isn't in the sort of mentioned in publications is the influence of pressure on degradation rates and on your model. And I'm wondering if you could comment on whether it's even possible to simulate hydrocarbon degradation at 100 atmospheres or 150 atmospheres.
Yeah, so the reason that we don't take it into account is because you can think about biological effects of pressure and adaptation to pressure, and you can think about how that's going to impact the substrate and the hydrocarbons. The hydrocarbons were sufficiently dilute that the pressure wasn't doing anything physically to keep them in solution or anything like that. They didn't exsolve when we brought them to the surface. That is, the gases didn't bubble out of solution. So there was no impact on the abundance or distribution of the hydrocarbons in any major way, except a little bit with methane having to do with gas hydrate. The physiological adaptations to pressure that have been observed tend to be at much greater pressures than what we're dealing with here. At 1,000 meters or 1,500 meters, there's not a lot of known adaptations. Those adaptations tend to happen as you get down to 3,000, 4,000, 5,000 meters. Organisms will really change their fundamental biology in certain ways. But at 1,000 meters, it's not so much. For microbes, different for macrofauna.
We have another question, please.
Michael Lange from German Public Radio. I have a question. Can you, from your work and from the work of others, tell when will the degradation of the carbohydrates go so far that we will have a concentration of carbohydrates that we had before the oil spill?
I don't quite understand. The carbohydrates?
I believe he means hydrocarbons.
Okay.
Oh, sorry for that.
Yeah. That's a very difficult question to answer because the hydrocarbon distribution before the spill was fairly patchy. That is, you have areas where you might have a little bit and other areas where you don't. So below detection and peaks that are above detection from various sources. Within, by October, after my third expedition, the water column, we could still detect hydrocarbons, but they were diluting substantially by that point. Extrapolating out from there, maybe a couple months to dilute out to being equivalent to the background levels for some hydrocarbons. Having said that, then there's pockets where things are going to get caught. The water column will mix, but the sediment will not. And so places where the oil contacted sediment and deposited into the sediment, or some of the unusual physical processes that may have rained waxes and other components of the oil out into the seafloor, you might expect to see those there for much, much longer. Beaches, beach sands, as we heard about in the same session earlier, are another place where things can accumulate. So the time scale is difficult because every location, every microenvironment is going to behave differently.
We have no questions from the web. We do have a question in the room.
Matt Kane, National Science Foundation. So Dave, has anybody calculated a mass balance of how much carbon was released total in all forms, including dispersant, and what the fate of that carbon is?
That's something that we've been working on, and different people have taken different pieces of the pie. It turns out to be a rather complex question because very few people look at all the different fractions of the gases and the oils. And so what we found is that all the measurements that were made, be they pre-spill measurements of the reservoir, they're all done differently, and none of them are entirely consistent with one another. And so a team of us has been trying to compile all that data and to get a complete mass balance and to move away from the barrels of oil, which doesn't include gas to oil ratios, which are normalized to standard cubic feet per barrel. It's really a bit of a mess to piece it all together. We're getting there. We have working values, but we're not quite ready to send them out yet.
Okay, we have a question from the web.
Okay,
From
please.
our Ustream chat, Lava Q wants to know, how long should the public be concerned with eating tainted seafood from the Gulf?
I don't study seafood, so I really cannot say what the, follow the advisories that the National Oceanic Atmospheric Administration is putting out.
If there are no other questions from the room or the web, let me ask one final question, and that is, and I think you began to answer that in an earlier response. You've developed an elaborate way of analyzing what the water's doing in this particular place. How much of a challenge is it going to be to adapt that approach to the next? There's going to be another oil spill somewhere. Who knows when and where? Not that we're wishing for it, and we got through a big one yesterday. We're all here today. But can you easily plug this in somewhere, or do you have to know the contours of the underwater space? And
Yeah, it's really going to depend where it is. If the next spill happens 10 miles from where this one happened, we got it pretty well understood, the basic processes. If it happens in the Arctic, in Alaska, offshore, then it's going to be a real challenge, and far more challenging than this situation. If it happens in the North Sea or off Brazil, then those are going to be very different situations as well. So it's not plug and play. We can't just take this and immediately apply it. You have to get out there and study, and the only reason we were able to do this is because we went out there and we were funded by the National Science Foundation, Department of Energy, to go out and collect samples and NOAA and start to look at what was going on, and then we could feed that back into what the Navy was already doing. Somewhere else, that would not be so easy.
so it's a time-consuming, energy-consuming bootstrap kind of operation each time.
Yeah.
And that it makes sense that you've got to tailor your analysis to the situation at hand. Well, I've been talking with David Valentine of the University of California, Santa Barbara. That's it for another ASM Live session. Tune in tomorrow for some more. Thanks very much.